So welcome to the Focus on Tumor Microenvironment and Metastasis seminar. This is part of the SPP Cancer Center seminar series, which is organized by Svasti Harisharan. And today we have two great speakers lined up focusing on cancer tumor microenvironments. I'm really excited to hear the talks today. Great, so it's now great pleasure to introduce Ashani Weeraratna to give the second presentation. So Ashi started her lab in 2011 at the Wistar Institute in Philadelphia. I think that's where we first met when I gave a talk there. Since 2009, Ashi has been a senior investigator at Johns Hopkins and is the E.V. McCollum Chair of Biochemistry and Molecular Biology at Johns Hopkins Bloomberg School of Public Health, and the Bloomberg Distinguished Professor of Cancer Biology, and also co-program leader, Cancer Invasion and Metastasis at the Sidney Kimmel Cancer Center. So very much a leader in the field. And in 2020, Ashi was the president of the Society for Melanoma Research. And you know, Ashi's work, I think, is, straddles, a few areas which are not often straddled. Okay, so in particular, she's very interested in the role of tissue aging and microenvironmental aging on and its impact on tumor metastasis and invasion and also response to therapy. Okay, so these are very, very important areas which I think are studied, and Ashi is really pioneering these areas. So I'll hand over to Ashi. Thanks very much, Ashi. It's great to have you to speak. Thanks.
Thank you, Peter. First of all, that was just an amazing talk by Shreya. I'm so excited to see that research, and I think you'll see why. Mine is going to be a lot more superficial because I wanted to give you guys some snapshots of different things that are going on in the lab. But I really enjoyed that talk, Shreya. So thank you so much. Thank you to Peter and Srasti for the invitation to speak here. And hello to all my old friends at SBP like Zev, with whom I hope to get a chance to speak later this week. So I'll start off by saying that, we are very interested in aging and how aging governs tumor progression. And the reason for that is because, of course, most of the people who get cancer are over the age of 50, and certainly the vast majority who die of cancer are over the age of 50. And as our population ages over time, we are going to see this as an increasing public health burden in the US and across the world. We've been very interested, of course, in the sort of systemic changes that happen that can drive this, such as changes in adaptive immunity and chronic genetic accumulation. But really what we have focused on is trying to understand what changes in the local tumor microenvironment as the host ages, and that works to drive changes in tumor progression. And so we've been interested in everything from the biophysical matrix to the immune microenvironment, and we focused a lot on fibroblasts and their impact on all of these different aspects. So the reason we focus on fibroblasts is because my lab studies melanoma, and a melanoma cell spends a lot of its time at the intersection of the epidermis and dermis, which is where it becomes malignant. We've wanted to know, as the skin ages, how aging fibroblasts can then drive the progression of those melanoma cells and make them even more metastatic. So some of the first early experiments that we did were to make artificial skin using fibroblasts from the upper inner arm, which has intermittent skin exposure of either people in their 20s or people in their 50s, and using a technique that was developed by Dr. Meinhard Herlin at the Wistar Institute. We made artificial skin, and the only parameter that we changed was the age of the fibroblasts that went into that skin. When you look at these skins histologically, what you find is that the melanoma cells that are made in skin reconstructs with young fibroblasts remain tightly nested at the surface of the reconstruct. Those that are made with aged fibroblasts, on the other hand, lose this sort of tight nested structure and start to invade into the dermis. We wanted to recapitulate this in animals, and so we used a melanoma model that was created by Martin McMahon and Marcus Bosenberg. And what Marcus has done is to backcross these animals, the C57 black 6 mice, take these tumors and create cell lines that can now be injected syngenetically back into either young or aged mice. When we do that, what we see is a fascinating set of differences. One of them is that these tumors actually grow much better in young mice. So we're talking 6 weeks compared to about 18-month-old mice here. And they grow much more slowly in the aged mice. However, when we look at metastasis, they metastasize far more effectively in aged mice than they do in young mice. So Mitch Fain in the lab wanted to know whether this was due to the fact that cells left the primary tumor in the aged mice more effectively, or whether they got to the lung and grew out more effectively in the aged mice. And so he did an experiment where he implanted tumors that were tagged in the skin of young and aged mice, and then he waited 3 weeks and examined the lungs. When he does that, he finds that the melanoma cells exist as these small single or double cell colonies in the lung. But if he waits just a few more weeks and takes the lungs at 5 weeks, what he sees is that in the aged mice, by 5 weeks, these are growing out like crazy, but in the young mice, they're remaining as these 2 or 3 or 4 single cell colonies. And that was quantifiable across several mice. So Mitch wanted to know what it was that was driving this outgrowth in the aged lung. And he went to a model where we used GFP tagged melanoma cells that were surrounded either by young fibroblasts or by aged fibroblasts. And he wanted to know whether the aged fibroblasts were secreting factors that drove proliferation of these cells. And indeed, if he looks at the melanoma cells in these cultures, they grow much faster with the aged lung fibroblasts, which is in direct contrast to skin, where aged skin fibroblasts slow down the proliferation of melanoma cells quite dramatically. So what we wanted to know was whether this had implications for tumor dormancy. So we used some of the markers of tumor dormancy that our friend Julio Guiraguiso at Mount Sinai has identified. These include things like AXL, NR2F1, P27, P21, et cetera. And when Mitch takes melanoma cells and treats them with conditioned media from aged fibroblasts in the lung, as compared to young lung fibroblasts, he finds that he can drive these melanoma cells into a dormant, that these melanoma cells have a dormant phenotype in the young, and he can drive them into a less dormant state in the aged. And when he looks at markers of proliferation, he sees that, again, the same is true, where the markers of proliferation are dramatically increased by conditioned media from aged lung fibroblasts. Mitch has now done proteomic analysis of skin versus lung fibroblast secretomes, and he's identified several members of the Wnt signaling family that have an impact on this. And so you heard about Wnt5b from Shreya just a few minutes ago. We also find non-canonical Wnt signaling is absolutely critical in the aged skin, but needs to be shut off for these tumor cells to grow out in the aged lung microenvironment. And so we've been looking specifically at factors like SFRP2 and SFRP1. SFRP2 plays multiple roles in therapy resistance in melanoma and an aged microenvironment, as well as drives angiogenesis. I'll tell you a little bit about that in just a second. And this interplay between these two family members that have opposing effects on canonical and non-canonical Wnt signaling are driving a dichotomy of growth versus dormancy in the aged lung microenvironment. So right now, our model for the work that Mitch is doing is that Wnt5a high cells will migrate from either young or aged tumors, and they get to the lung, and in the young lung, they stay dormant. Wnt5a is critical for the initial dissemination of these cells and critical for their survival in the young. But in order for these tumor cells to grow out, Wnt5a has to be shut off. That is affected by SFRP1 in the lung microenvironment, and then those tumor cells can start to grow out very rapidly. Mitch has also shown that the immune microenvironment in the lung changes dramatically, and we see a profile of increased MDSCs, increased Tregs, and an overall increased immune suppression in the aged lung that also contributes to the outgrowth of these tumor cells in the aged lung. I wanted to focus a little bit on some of the matrix changes. So over time, we found that aging impacts multiple factors in the tumor microenvironment, from tumor dormancy, which I just touched on, angiogenesis, which you'll hear about in a second, metabolism, and the immune microenvironment. And so because I knew Shreya was speaking and was going to talk about ECM and matrix changes in the tumor microenvironment, I wanted to share a little bit of some of the newer work that we're doing that is based on work from a couple of years ago where we looked at changes in the aging ECM. So Manpreet Kaur, who was a graduate student in my lab, who's now at Penn in Arjun Raj's lab, she looked at the density of collagen during aging, because what we would see all the time is that when we made our skin reconstructs, the collagen in the aged skin reconstructs looked a lot looser and lacier, just even to the sort of visible naked eye, than that in the young microenvironment. So Aman performed 2-photon microscopy. She quantified the collagen in the young reconstructs as compared to the age reconstructs. And just to be sure that this was not a factor of the fact that the cells were invading and showing up the collagen, she also took normal mouse skin from young and aged mice. And again, when she does 2-photon microscopy on the skin from normal non-diseased mice, she sees that the collagen in the aged mouse skin bundles up into these fibrils with a lot of space in between, whereas in the young mice skin, the collagen is very diffuse and tightly networked. She wanted to know what was driving this, and so she turned back to the proteomics. And one of our most highly secreted proteins in the tumor microenvironment, in the young fibroblast secretome, was a protein called haplin-1. And haplin-1 was critical for stabilizing aggregates of proteoglycan monomers with hyaluronic acid. And hyaluronic acid is critical for maintaining collagen and elastin interactions in the skin. And as you age, the reason you get wrinkles is because we lose hyaluronic acid, which I can never say properly, and those collagen and elastin links start to break down and the skin starts to wrinkle. So Aman decided to explore the role of haplin-1 in the young and aged microenvironment. And what she saw was that, that if she takes young fibroblasts and allows them to form matrices, so right here what she's doing is, I'm so sorry, this is on an automatic timer for some reason. And when she takes fibroblasts, she treats them with ascorbic acid. She allows them to lay down a matrix. The fibroblasts, when they are young, lay down a matrix that has fibers in multiple different directions. If she now decreases haplin-1, she knocks it down in the young fibroblasts. She takes them from this multi-angular network into a more linearized network. If she looks at, and if she adds back haplin-1, she can reconstitute that sort of network cross-linking in the young fibroblast matrix. If she now does this with aged fibroblasts, what she sees is that they start out already very linear. And if she treats them with recombinant haplin-1, she can knock that matrix back up, cause a lot of these cross-linked fibers, which you can really tell by the difference in the colors that you're seeing, because the light is refracting off them at different angles, making, and those are assigned different colors. So Aman then took that work further. She showed that when you have tumor cells that are existing in matrices like this, when they're in a young matrix that's very tightly cross-linked, they don't invade. When they're in an aged matrix that has more of these linear structures and less cross-linked, those tumor cells invade much more rapidly. And she could manipulate haplin-1 to drive that invasion. I'm not showing you all of those data because they're already published, and I'm just using this as a background for some of the newer work we're doing. Brett Ecker in the lab, who was working side by side with Aman, also found that the ECM could affect lymphatic vasculature. So what Brett found was that in the young, lymphatic vasculature and the lymph node, the ECM that the fibroblasts laid down along those vasculature made a very tightly cross-linked ECM. And then tumor cells were sort of kept in the vasculature directed to the lymph node. However, in the aged mice and aged patient samples that we looked at, what we found was that as the ECM started to break down, it was creating a lack of integrity in the lymphatic vasculature, allowing tumor cells to escape, some before they ever even got to the lymph node. And so Brett concluded from that work that aging actually affected the route of metastasis. That's a clinical observation in melanoma, where younger patients actually have more lymph node metastases than aged patients, but aged patients have far more visceral disease and die more frequently of that disease. And so he also found that haplin-1 could reconstitute that ECM around these vessels and give back integrity in the aged mice if he reconstituted it. So Gloria Marino, who is a graduate student in the lab, decided that she would pursue this and look to see how this affected angiogenesis and blood vessels. And so what she has been doing, I apologize, my dog is annoyed that I'm talking to you, so you might hear some whining and growling. But what Gloria has been doing is she's been looking at blood vessels, so endothelial vessels rather than lymphatic vasculature in the aged microenvironment as compared to the young. And what she finds is that blood vessels in the young microenvironment are surrounded by very dense collagen, but in the aged microenvironment, there's a lot less collagen. And of course, this is reminiscent of what I just showed you with the young and the aged mouse skin. So Gloria took mice into which she implanted tumors in either aged or young mice. We have seen multiple times that there's far more angiogenesis that goes on in the aged mice. And so Gloria showed that yet again. But now she showed for the first time that if she treats with recombinant haplin-1, she can actually decrease angiogenesis itself. And we're still trying to figure out the mechanisms of that. But what was interesting about these blood vessels was that in the aged mice, the blood vessels, although they were more abundant, were much less likely to stain for V cadherin as they were in the young mice or the mice that had haplin-1 reconstituted. And so this suggested to us then that the blood vessels in the aged mice might be leakier. So to test this hypothesis, what Gloria did was to take fibroblasts, allow them to lay down their matrix, and then she laid endothelial cells on top of those matrices. Her hypothesis was that if the matrix in the young mice was nicely cross-linked and stable, then the V cadherin connections between the endothelial cells should be stabilized by the endothelial cell connection with the matrix. And then she also hypothesized that as those fibroblasts age and they lay down these less cross-linked matrices, that would disrupt the endothelial cell to ECM interactions, which in turn would disrupt the endothelial cell to cell interactions. So she performed this experiment. These are HUVECs that are laid down on matrices from either young or aged fibroblasts. And when she stains for V cadherin, you can see that in the young, in the endothelial cells on the young fibroblast matrix, they have very nice V cadherin zippers, if you will. But those same cells plated on an aged matrix lose these cell-cell interactions. If she now brings haplin-1 into the mix and asks what happened with haplin-1, what she sees is that if she knocks down haplin-1 in the fibroblast before allowing them to lay down that matrix, she can disrupt the cell-cell interactions with V cadherin. And if she reconstitutes haplin-1 to the aged fibroblast before they lay down their matrix, she can now restore V cadherin junctions in these cells. So what that said to us was that an aging matrix can disrupt these endothelial cell-cell interactions and could potentially impact permeability. And so to examine that, what Gloria did was to collaborate with Karen Eisinger Matheson at UPenn. and perform a trans-epithelial resistance assay. And so the way these are performed is that we allow the fibroblasts to lay down their matrix on an electrode. We plate the endothelial cells on top of that, and then we measure electrical current that goes through those cells. And so if there is resistance, that means these barriers are very tightly joined together. And if there's no resistance, it means that the barriers are not intact and molecules can just flow very freely between the cells. So what Gloria found was that when she plates endothelial cells on something like gelatin, they have a very nice, strong resistance that increases over time. If she plates endothelial cells on matrices made from young fibroblasts, the same thing happens. If she plates endothelial cells on an age matrix, there's no resistance and no increase in resistance over time. But now she gives those fibroblasts recombinant haplin 1, she can increase the resistance of the endothelial cells, so knotting back up those boundaries, which fit very nicely with her V-coherent work, both in vivo and in vitro. So to sum up this project, basically, we've been looking at how aging impacts angiogenesis, and I've shown you mostly data on the vascular permeability. It's been known for a while that aging impacts endothelial. I'm so sorry. It's been known for some time that aging impacts angiogenesis and endothelial cell function. Ironically, although we see more angiogenesis, we see a decrease in things like VEGF. You see endothelial cell senescence. That's all been shown by many other groups before. And something that's been very interesting, though, is that we see less VEGF, but more angiogenesis. That's due to this SFRP2 factor that I mentioned earlier, which is increased during aging and drives a high level of angiogenesis in tumors in aged mice. This means that patients who are older actually have a worse response to Avastin, which is a drug that is meant to target angiogenesis, but the way it targets angiogenesis is to inhibit VEGF. And we've just recently published this, which is why I'm not spending more time on this right now. The work that Gloria has been doing suggests to us that tumor cells that are in either trying to get into a blood vessel or trying to get out of a blood vessel are going to have a much tougher time in an aged microenvironment where those endothelial cells are held together nicely by a firm matrix around them, whereas those tumor cells in aged blood vessels are going to have a much easier time because they'll be able to escape through what are essentially leaky endothelial walls. What I'd like to do is end with one quick story that we just recently published, but it's a story that I love because it really it really shows how we can target age-related molecules to overcome resistance to therapy in melanoma. And so this is a story that was started by Gretchen Alisea when she was a graduate student in my lab. She's remained on as a postdoc in my lab, although her work is now taking a different direction. And what Gretchen has noticed was that if she gave aged fibroblasts sugar in their media, they would make a lot of lipids. And in general, when she treated melanoma cells with conditioned media from aged fibroblasts, they too would either make or take up a lot of lipids. She performed lipidomics and she found that the same lipids that the fibroblasts were making in the aged fibroblasts were making were the same lipids that were being taken up by melanoma cells. She wanted to know what was driving this. And so she sort of hunted around for a while. She identified this transporter, FATP2. If she takes melanoma cells and exposes them to age-conditioned media, she sees an increase in FATP2. If she looks at her tumor cells in aged skin reconstructs, they have more FATP2. If she looks at melanoma cells that are implanted in aged mice versus young mice, she sees an increase in FATP2. And finally, when she looks at melanoma patients over the age of 50, have far more FAP-P2 than those under the age of 50. So Gretchen wanted to know if she targeted FAP-P2, could she decrease lipid accumulation? And so she knocked FAP-P2 down in her melanoma cells, exposed them to age-conditioned media, and showed that now the melanoma cells could not take up as many lipids. And then she also treated with lipofumata, which is a drug that targets melanoma cells in the presence of age-conditioned media, and showed the same thing, that if lipofumata was present and FAP-P2 was knocked down, the melanoma cells did not take up as many lipids. She wanted to know what that meant for melanoma patients. And one of the things we know is that metabolism affects the way tumors respond to targeted therapy, specifically therapy targeted against the BRAF oncogene. And when she looked at patients who had been treated with inhibitors to the BRAF MEK pathway in melanoma, those that did not survive and had a really a shorter survival time were ones that had very high level of FAP-P2. Now, we didn't have a lot of samples, so Gretchen decided to try to manipulate the system in vivo and see what happened. So the first thing she did was to create a DOX-inducible knockdown of FAP-P2 in her melanoma cells. She implanted those into mice. And what she saw is when she implants them into young mice, the FAP-P2 has no effect. So the tumors grow whether or not they have FAP-P2. If she treats with the BRAF MEK inhibitors, which is this blue line, the control tumors go down, and then after a while, the tumors start to grow again. And if she does this in the presence of a FAP-P2 knockdown, they do exactly the same thing. However, when she does this in age mice, it's a very different story. So first of all, treating age mice with BRAF MEK inhibitors has almost no effect on the tumors. They stop growing for a little bit, but then they continue to grow right through it and look just like the controls. However, now if she silences FAP-P2 and then treats with the inhibitor, not only can she cause those tumors to regress rather than just stop growing for a little bit, they regress and they stay gone. So this was super exciting for us. This is one of the targets we're actually driving into clinical trials. We're testing some of the inhibitors right now. And so this is a super exciting project for us. And so I will just finish here because I'm probably well over time by now. And just again say how very important it is to consider age as a factor when treating patients and designing preclinical studies. I hope I've convinced you that the aging matrix is important in driving invasion. Angiogenesis is increased during aging. There are changes in metabolism that can drive resistance to therapy. And of course, all of these go hand in hand with increases in metastasis in aging patients. We're now translating our data into different cancers. We recently just got back the first in vivo data from pancreatic cancer, and it's really fascinating and exciting. So these are the people who do the work. This is a group that moved with me from Philadelphia, and these are all of the new people who've joined us. Collaborations across the street, across the country, and across the world. I thank many of my wonderful collaborators and colleagues for their contribution to this work. And this is my favorite aging study, who's my daughter, who's now a 16-year-old and driving, which is super scary. And I'll end with reminding you all that we cannot let our guard down when it comes to equality in science of people earning STEM degrees. Black, Latina, and Asian women hold, all told, a little less than 10% of bachelor's degrees in STEMs, let alone PhDs. I recently wrote this piece with my friend Danita Brady. We have a table in there that has some action items if you're interested in implementing them in your department and in your environments. This is something that I think, you know, we all require, we all need to play a role in increasing diversity in the biomedical sciences. And with that, I'll stop and take any questions.
Thanks, Ashi. That was fantastic. And I just want to say, I know that Shreya is also a big advocate for racial diversity and in the scientific environment, which is something which I know we all agree is very important. So thanks very much. Before I hand over to Svasti for questions, I have a bunch, but I'll start with just one. So you referred a number of times to aged fibroblasts, and when I think of aged fibroblasts, I think of two possible things, and maybe others. Obviously, one is cellular senescence, and then the other one is activation of fibroblasts. Fibroblasts can take on a myofibroblast type phenotype where they express SMA, smooth muscle actin, which is certainly not a feature that we normally think of as senescence. So there's at least two possible phenotypes there. So I'm wondering which one of those or something else do you think is important in driving the phenotypes that you're looking at?
Great question, Peter. So first of all, I will say that when there's tumor present, the SMA levels look the same between young and aged. There's not a whole lot of difference. Without tumor cells present, like in things like mouse skin or our artificial reconstructs, we have seen that there's a slight increase in SMA in the aged, but not in the young. And once tumor cells are there, that all works itself out. In terms of senescence, there's definitely more senescence in general among the aged fibroblasts. What's interesting is we're now doing some sexual dimorphism studies to see the difference between male and female. And we see that there's far more senescence in young female fibroblasts than in young male fibroblasts, but they both trend to increase as the fibroblasts age. What I will say is that we've looked at the SASP in these fibroblasts, Judy-Campisi's secretory phenotype, and we see some overlap, some of the key players, IL-6, IL-10. However, we don't see that all of the molecules are the same. So the damage-induced SASP is very different than the age-induced SASP.
Right. Okay. Yeah, I think dissecting apart those two is going to be a really
it's going to be brutal
question. Anyway, I'll hand over to Svasti. So thank you very much.
Thank you, Ashi. That was a great talk. Obviously, lots of questions, but thank you so much for the shout out to all of us being aware about diversity in science. I think it's so important, or the lack thereof. So I'll start off with Zev's question for you, because it's sort of really the most targeted, I think, to your talk. What he wants to know what controls the expression of SFRP1 to enable its spatial localization, and whether dormancy in other models is dependent on SFRP1, and whether HAPL-N1 expression specifically is inversely correlated with SFRP1. I can do the questions one by one again.
No, it's fine. So Zev, we are not sure. We've been looking at methylation of the different SFRP molecules. Nothing is coming up as abundantly clear yet. So we see SFRP1 is increased in age lung, but not in age skin. SFRP2 is increased in age skin, but not in age lung. And we're not quite sure yet what controls that. We're assuming at some point it's going to be some crosstalk between the tumor and the lung fibroblasts. We have not looked at dormancy in other models for SFRP1 specifically. We have looked at Wnt5a, however, and Wnt5a does drive dormancy in breast cancer. That's work we've been doing with Julio, and also DKK1 is another Wnt that drives the same level of dormancy in breast cancer and melanoma. And then the other question was HAPL-N1 and its relationship to SFRP1. So, oh my gosh, this dog. So HAPL-N1 is, we haven't looked at the relationship yet between HAPL-N1 and SFRP1. I'm not sure that we expect a relationship between them, except for the fact that we've just now found that it can affect angiogenesis, so it might have a relationship with SFRP2.
So going back to your last project, actually, that you talked about, one of the questions was, what do you think is the mechanism underlying this lipid transporter affecting the metabolism of small molecule inhibitors like the MEK inhibitor, et cetera, that you use?
Right. So we don't think that it's actually affecting the metabolism of the small molecules themselves. Initially, we thought that might be happening because we thought they might be compartmentalizing in lipid droplets, but that's not the case. Because phospho-ERK is not really affected when we treat with these inhibitors. What we do think is happening is that the FAP-2 is able, and the accumulation of lipids affects mitochondrial metabolism, which ultimately affects, whether or not the cells respond to the therapy.
So I know Peter has another question about Haplan-1, so I'll let him get that in, but then I do want a little bit of time for questions for both of you. I think would be a more interesting dimension, but go ahead, Peter.
Okay, thanks. So I'm sure you're familiar with Vera Gorbanova's work on hyaluronic acid in naked mole rats. So it's the obvious question about, have you looked at Haplan-1 in naked mole rats? Is it?
No, we haven't yet. We have not. We haven't had the chance to do that yet. We have been looking at HA, though, in melanoma and in skin, and how manipulating Haplan-1 is affecting HA.
Okay.
And so certainly we see that there's an increased release of HA in the absence of Haplan-1. So we really are thinking of Haplan-1 as the knitting needles that's sort of knotting in the HA with the collagen and elastin.
Yeah, which I think that would fit very much with Vera's view of the high molecular weight HA being tumor suppressive as well. So yeah, okay, great. Thanks, Ashley.
Thank you.
Can I ask Shreya, can you also turn on your video and unmute yourself just so we can get the second part going? So I think one of the questions that really comes up listening to both of your talks, and it's come up in the question and answer session here as well, is it seems to me that, yeah, you do see this effect of the aging microenvironment in the lung, right, as Ashi showed, and also with your decellularized matrices. But it's interesting to me to ask, how does the aging microenvironment of the primary site that the cancer grows in affect how the cancer cell then responds to the aging microenvironment in the distant organ that it then ends up in, right? So looking at Ashi's talk, for example, it does make you wonder whether the aging cells are more dormant, but because of that negative feedback signaling they're getting at their primary site, when they migrate to a distant organ, they're more able to respond to cues and establish themselves as colonies. So for both of you, Ashi and Shreya, one, what do you think of that? And two, maybe especially for Shreya, how could you model that?
So I guess I'll start. So the way we've been thinking of it is that there's a signaling profile, we call it phenotype switching in melanoma. That's a grow versus go, right? So there's signals that make them grow and there's signals that make them go. And unlike other cancers where those might go hand in hand, in melanoma, it's very different. Like you need to shut off one set of signals in order for cells to invade, and then shut off those invasive signals in order for them to grow. And so rather than thinking of it as, it's losing some restriction, which allows it to invade, that restriction in the primary site, we think, allows it to invade whether it's young or old. And then when it gets to the age microenvironment, or whether it sits in that young microenvironment for some time, the changes that are happening in the age microenvironment eventually shut off those dormancy / invasive signals and survival signals, and allow them to grow out. Because the cells that are growing out are much more delicate, if you will. They have all of these markers that the immune system can recognize. A lot of them are markers involved in melanin production, like MART1 and GP100, and those act as red flags to the immune system. But take that into account with the fact that the immune system is also being, suppressed, if you will, and now those cells can grow out in an age microenvironment.
So I'll add to that, as from an engineer's perspective, at the primary tumor microenvironment, there's a growing tumor experiences different mechanical forces than a tumor that's ready to invade. And oftentimes, and there's enough evidence in literature to support this, that, with the growth comes hydrostatic pressure or compressive forces and tensile forces that change the phenotype of the cells enough to, like Ashi said, to stop and go from grow to go, or in specific cancers to balance between those two signals. And so that from an engineer's perspective is tunable, right? So we can create like stiffness and compressive forces that can mimic either of these conditions, and whether that's at the primary site or in a metastatic organ. There you go, Ashish. She just wanted to be on your lap.
Sorry, she won't stop. So
I'm gonna let Peter sneak in a question for the two of you as well, and then there's one last question, and then I'll let you go.
Yeah, so I know from conversations I've had in the senescence field that people are very interested and are trying to incorporate, for example, senescent cells into organoid type models. But generally, I think that's challenging because senescent cells behave very differently. They don't proliferate, for example. So I wondered. I mean, this is maybe a question of interest to both of you, but maybe more practically for Shreya. Shreya, have you tried perhaps incorporating senescent or aged stromal cells into some of your models?
So we tried to induce senescence with irradiation, and it really, very first pass pilot experiments, and we didn't do very well. But mostly, again, like you said, they don't proliferate, and so when they don't proliferate, there's just not much for us to work with. But we haven't thought about it seriously. And there's it is something that is of interest, but I don't think it's as non-trivial as some people seem to think it is. We've definitely attempted it and have had some difficulties trying to get it into a 3D microenvironment without additional sort of scaffolding support.
Okay. I mean, one thing I would want to comment on that, I guess. I mean, senescent phenotypes do vary. I think the IR-induced models are potentially, pretty extreme. It may actually be that with, some of the more physiologically aged models that that Ashani is working with, I think, they would be more physiologically relevant and perhaps also more tractable.
Or the DNA repair defective models that we were
right.
we do in my lab. And another question for both of you, I think, is how does treatment resistance into the primary tumor, within the primary tumor cells, say in the aging skin in your melanoma model, does that promote metastasis or inhibit metastasis or somehow change how the cells respond to the ECM in the distant organ?
great question. So, what I will say is we find that a lot of the same signaling pathways that drive resistance are also the ones that drive invasion. So whether it's kind of a chicken or egg thing, are they resistant and then becoming invasive? Are they invasive and then becoming resistant? And then there's this whole additional phenotype that we've seen that we published on several years ago, we call pseudosenescence. And what happens is we treat cells with BRAF/MEK inhibitors or irradiation, the melanoma cells themselves, and they undergo what looks like senescence. They stop growing, they have chromatin marks, beta-gal, PML bodies. They look just like senescent cells, but they become more invasive. And when you put them in animals, they go to the lungs, they grow out. You can overlay markers of proliferation and beta-gal, and they have both Ki67, beta-gal, chromatin marks. So you can drive that phenotype, that invasive phenotype, with drugs, yes. But I also think that if they have those signaling pathways, they're going to be more likely to be resistant as well.
Okay, so if there aren't any more questions, I just want to remind everyone that the next seminar in this series, also really exciting, Alana Whelm and Alice Serrani, there it is, who work on PDX and cancer modeling, so a different perspective to what we saw today, but I think equally exciting. So please join us on May 10th, and then I'll let Peter wrap up and thank both our excellent speakers.
Can I ask Ashi a parting question?
Yes.
How soon before I start seeing Haplin 1 in my Cetaphil face cream, Ashi? Like that's what I want to know.
That's the low-hanging fruit, right?
Thanks very much, Ashi and Shreya. Really enjoyed your talks, and also to Svasti for organising the series. So I know that Ashi and Shreya are going to be having a chat with some of our trainees at 11:30, I think, Pacific time. So we also, very much appreciate your time there. I think it's, it's obviously really important for trainees to get the chance to speak to, scientists such as yourself. So thank you. So with that, thanks very much for joining, everyone, and see you next time.